Welcome all to this video lecture. I am John Milton, working as an assistant professor in the Department of Microbiology, St.Joseph College of Arts and Science, Cuddalore Autonomous. So, in this video lecture, we are going to discuss about the red rot disease of sugarcane from the subject of soil and agriculture microbiology. So, sugarcane is one of the important commercial crop to the farmers as well as to the millers. So, some problems, some diseases to the sugarcane will affect both the farmers as well as the millers who are running the industries, sugarcane mills. Usually, the loss to the farmers is due to the poor utilization inputs, gaps in the field, a low yield, poor ratoons. So, the ratoons are nothing but the emerging plants after the harvesting from the field, that is ratoons. Poor ratoons, poor quality seeds. So these are all the things. Because of these reasons, the farmers get loss. The loss is to the millers, that is due to poor juice quality, poor sugar recovery, and less bagasse, increase in molasses. So these are the reason to create the loss to the millers. So both farmers as well as the millers are affected if any diseases or any damages or any problems are found with the sugarcane. So both will also result in the loss to the nation also. So the distribution of the red rot disease is it is found in India as well as in the world wherever the sugarcane is cultivated. It is one of the serious destructive diseases. It causes severe loss to the farmers. The sugarcane is the native of India, but the disease is first reported in Java in the year of 1893. So after that, it has been reported worldwide wherever the sugarcane is cultivated, including Cuba, China, in like this area. So it was first seen in India in the year of 1901 near the Godavari Delta regions of Andhra Pradesh. It occurred in eastern Uttar Pradesh and northern Bihar in the year of 1939 and 42. In between this period, it will cause severe loss to the farmers. So after that, much importance are given towards this disease and control of these diseases. So the accepted name red rot was given to this disease was by Butler. The causative organism which is causing this disease is Calectotrichum falcatum. The mode of entry, how the organisms enter the plant and causing diseases. The planting materials may harbor the fungus and this transfer the diseases from season to season. The planting materials, the sets, so that may harbor, that may having the pathogenic organism, pathogenic fungi or its a spore, and that is the main reason for spreading the diseases. The fungus also persists in the soil after harvesting in the diseased parts like leaves, stem portions. That is also another reason for introducing for emerging the infection to the field after harvesting. So the primary infection is by infected seeds. Seeds is the cut portion of the sugar cane which is used as seeds. That is the seeds. And the secondary infection is through the irrigation water cultivation tools like knife and other tools which are used for the plowing as well as the wind. So after conidia settled on the leaves, they germinate and invade the leaves and establish their growth on the leaves and other parts of the sugar cane. So the stem infections take place by insect borers. So in the case of the stem, some insect bores, and through these pores, the fungus can enter and cause disease. So this is the seeds of the sugarcane, so through which the organisms may enter. So the pathogen also enter the healthy seeds through the cut end from the soil, causes early infection of the shoots. Sometimes you may choose the healthy sugarcane as for the seeds, but the knives as well as the soil may having the pathogenic fungi. It may enter through the cut wounds. It may happen sometimes. So what are the symptoms associated with the red rot disease? So the symptoms usually appear after rainy season. The affected can exhibit a leaf color change from green to orange and then yellow in the third and fourth leaf. So in the third and fourth leaf, the color change may occur, usually orange to yellow in color. So the later infected can shriveled and the rind become shrunken. So the external symptoms appear only after 16 to 21 days after infections and become dried. So the cane light in weight and easily broken. So the sugar cane that is usually the weight loss will occur and it can be broken easily. So that is one of the symptoms. The main symptom is the reddening of the pith with clear white area. That is the main symptoms, reddening. That's why it is called as red rot diseases. So you can see the pith which where the red color is can be seen. The pith cavity filled with the grayish mycelium. So the pith cavity will be full of the grayish mycelium. You can see in the pictures the grayish color inside the pith, that is the fungal growth. So the juice bad odor emissions, the bad odor, especially the alcoholic smell will be emitted from the juice. So that is due to the conversion of the sucrose into glucose, then the conversion of the glucose into alcohol by the fermentations carried out by the fungi. That is the main reason for the emission of the alcoholic smell, and that is one of the main symptoms for this disease. So the loss in sucrose is at usually about 33%. So automatically there will be reduction in the recoverable sugar in the factory because of the reduction of the sucrose in the sugar cans. So how can we control the disease? The crop should be thoroughly inspected, and if any infected plants are there, that should be immediately burned and disposed. So the waterlogging conditions should be avoided because the waterlogging conditions, the water may act as a transmitting agent of the diseases. So the waterlogging conditions should be avoided in the field. Otherwise, the next plant where the water goes can results in the emergence of the disease. So prior to sowing, the sets should be thoroughly inspected. If we found any kind of symptoms like reddening of the pith or any kind of shink or lightweight, if we see any kind of symptoms, that should be avoided. That the sets should be avoided. That should not be used as a seed. So after harvesting, the plant debris should be destroyed because there is a chances of existing of the fungus or fungal spores in the leaves portions. So usually that will be practiced by the farmers after the harvesting. The crop rotations of 2 years should be practiced. Suppose if the field is found with the diseases for the complete removal of the pathogenic organisms from the field. Especially the grain manure can be cultivated. That may enrich the fertility of the soil also. So the sowing of the susceptible varieties like Co 312, Co 3331, Co 213, like these varieties must be avoided. So because there is possibilities of occurring the infections with these varieties or more. So the resistant varieties can be selected and can be cultivated to escape from the loss due to the diseases, and the resistant varieties, some of the resistant varieties are given here, Co 453, and some varieties are given. So the set treatment can be done. So the carbendism, the chemicals are used at the rate of 2.5 gram per liter, and in this water we can immerse the seeds for 30 minutes. That will control the growth of the fungus. That will be effective. After that we can sow the seeds. So the hot water treatment can be practiced. So if the water is in the temperature of 52, we can immerse the seeds for 8 hours. Suppose if the water is at the temperature of 54 degrees Celsius, 6 hours is enough. So sometimes hot water hair treatment can be given. The hot air with temperature of 54 degrees Celsius can be passed over 6 hours towards the cells. That also will eradicate the fungus. But the hot water treatment and hot air treatment usually rarely applied to remove the pathogenic organisms from the seeds, because it may affect the germination efficiency of the seeds. So these are rarely practiced. So the foliar applications can also done with the chemicals bavestin at the percentage of 0.1 or baniate at the percentage of 70% by 2 to 3 times at the interval of 10 to 12 days may be tried to control the spreading of the diseases. So these are the way of controlling. So thank you for watching this video. Thank you all.